The young sea squirt is a fascinating animal. It has a tail to swim around, yet this swimming larvae stage lasts only a short time. They are not capable of feeding. Soon, they settle to the bottom and cement themselves headfirst to the spot where they will then settle. Now an amazing transformation begins. It absorbs its own tail, its primitive eye, and its spine-like notochord, and finally it absorbs its own rudimentary brain, so its cerebral ganglion. It does not need this superfluous brain material anymore, and instead it develops its digestive and reproductive organs. So having a brain means a certain energy investment. But aren't there other ways to rebalance energy resources? In this symposium, we will focus on the role of sleep for the brain. This part will focus on the connection of sleep and the brain in the first 2 decades of human life. From the perspective of a sleep researcher with training in biology, I will present this interdisciplinary approach, and I cordially thank the organizers for putting together this program. I am grateful for the multiple collaborators who were involved in the parts of science that I will show today. Those who contributed substantially are labeled in bold. Also, I'd like to thank our funders for making this all possible. And here you also see my contact information and our website. Across the first year of human life, total sleep duration decreases by 8 minutes per month. Why does sleep change so much? This is visible at the level of sleep behavior, but not only, as we will see later, also major changes happen in the sleep neurophysiology, so in the EEG. I'd like to discuss with you today the drivers that are underlying this change. Since the 60s, the knowledge exists that sleep composition undergoes developmental dynamics. These dynamics in childhood and adolescence are not comparable to the relatively minor dynamics that then happen again in adulthood. Based on these observations, the authors proposed that sleep possibly plays a role in stimulating neuronal maturation. Today, we have a more mechanistic understanding of how sleep behavior is linked to neuronal activity. In general, there are 3 different primary stages: wake, non-REM sleep, and REM sleep. They can be quantified on the level of EEG, and you see here also EMG, multi-unit activity, and cellular spiking activity. While waking and REM show obvious differences in muscle tone and eye activities, non-REM sleep is characterized by very typical large waves that are slow in frequency. That's why these are called slow waves. Those slow waves, they coincide in particular activity with the layers below. So there are states of on and off activity, meaning that all fire or all are silent at very synchronized time points, and there's an interchange of this firing and silent activity. Slow waves are interesting because they undergo major dynamics across childhood. So here you see the slow wave amplitude, and similar dynamics exist in slow wave activity quantified in microvolt square, which we will use later. So here again there was a link proposed to brain function, such that neuronal maturation and sleep are closely connected, and this is in the 1980s. So here particularly the author proposed that the pruning period, so the decrease of slow wave activity across the 2nd decade, would be linked to processes of schizophrenia. Yet in the last 10 to 15 years, we have indeed gained more understanding of how sleep and neuronal processes are linked. 2 relevant hypotheses exist, and I will mention these concepts only briefly today. The first is the shy hypothesis. Its concept is that wake represents a frame for synaptic buildup across our waking times throughout the day, whereas sleep represents a frame for rebalancing or downscaling synaptic strength. So the purpose of this is to make energy resources available for synaptic plasticity processes that underlie learning. The second hypothesis relates to brain myelin. Here, sleep represents the frame for myelin maintenance, such that in the very extreme case of sleep deprivation, the outest layer of the myelin of a neuron detaches from the node of Ranvier and retracts into the lower layers. Both hypotheses are supported by increasing research on animal, computational, as well as human models. Now we know that major changes are happening in the brain across childhood. One example in gray matter is that there are major changes in synaptic density and their interconnectedness. So this maturation of gray matter anatomy also undergoes a spatial dimension. So there's a local aspect of this maturation, and it goes in a pattern from back to front. So we wondered, is this anatomical maturation reflected in sleep? We therefore conducted a well-controlled study in healthy children in Switzerland and the US. And this was during the period where Reto Huber was my mentor and Monique Le Bourgeois. And we assessed high-density EEG scheduled to habitual sleep in 55 healthy children between 2 and 20 years. We did standard pre-processing of the first 60 minutes of non-REM sleep stages 2 and 3. And what you see here is a frequency of slow effectivity, so 1 to 4.5 hertz, displayed in a color-coded manner. So red means highest slow effectivity and blue means lowest. So the goal here was to identify whether there are developmental dynamics of local slow effectivity. And therefore we used individually color-coded maps that would, by the way, be the best EEG proxy for deep sleep, also from a behavioral perspective. So these results showed that there was the same back to front pattern across childhood and adolescence, similar to gray matter anatomy. And that would result in the frontal maximum visible in adults that has been well known until the day of publication. So we wondered, is there any functional relevance to this local slow effectivity pattern and its development? So next, we tried to capture whether the regions in transition have also more slow effectivity. We assessed 63 children and adolescents between 2 and 26 years of age and computed their slow effectivity maps. We then defined regions of interest based on Brodmann areas, and we quantified regions of interest like language, cognitive control, or simple and complex motor. And indeed, we found that the more mature the slow effectivity location, so on this back to front gradient, the higher were also the motor skills. And this was a comprehensive assessment with the Zurich neuromotor assessment, including, for example, sequential finger tapping, tasks, and reaction to an oddball paradigm. And they showed proximity in a cluster analysis. So thus, slow effectivity topography shows some relevance in behavioral functionality. We then took it one step further and explored whether the maturation of specific skills is predicted by the slow effectivity maps. And we found indeed that slow effectivity topography, so the map, matured earlier than the behavioral skills and also earlier than the gray matter dynamics. But as in any study, this is very much simplified. So, for example, not only the cortex is relevant, of course, also white matter. The primary content of white matter is myelin, which is to some degree quantifiable with McDespot MRI. So this is a model we used that is based on a two-pool microstructural model, and it has been used to track neurodevelopment and neurodegeneration, and it has been histologically correlated to myelin content. It is known that this measure of myelin water fraction increases rapidly across especially early childhood, and we wondered whether this is also reflected in the sleep EEG. We thus conducted a longitudinal study with preschool children at a baseline assessment, and we followed them up 3.5 years later. We were interested in myelin-specific MRI in natural sleep. Therefore, we adapted the scanner, as you can see, and we waited long evenings for those children to fall asleep. This was a collaboration with Sean Dioni and Monique Lebourgeois. Just very briefly, those results showed that the slow wave activity maps predicted later myelin outcomes, and especially this was visible in the whole brain content and the content of superior longitudinal fascicle. Now, from a translational perspective, the overarching question remains, why and how does this matter? In cats, rodents, and flies, chronically disturbed sleep in the developmental phase leads to consequences in later life that are visible at the neuronal and behavioral level. So how can we approach this in humans? We conducted an experimental study design where we restricted children to 50% of their sleep compared to a baseline night, and the sleep restriction was performed via a delayed bedtime. We performed in both conditions high-density EEG, and we conducted MRI once. The results showed that local slow wave activity increase was happening in posterior regions. So this was very much unlike adults, where this increase due to restricted sleep happens frontally. So these results suggest that those developing brain regions are more at risk through sleep deprivation. Further, we found a link to myelin content in the optic radiation, which was not in the same region that showed increased slow wave activity following sleep deprivation, but in adjacent regions. Let's recapitulate. Sleep is a physiological phenomenon at the cellular level, which spans the full range to the behavioral level. Sleep deprivation affects the EEG and also behavioral performance. So now we wonder, are there other interactions on the level of body periphery? So far, we have discussed the sleep-brain axis. Due to recent attention and novel insights in the gut bacteria, the gut-brain axis comes into focus. One example from mice that were raised without gut microbiota. These mice show altered HPA axis functions, so their stress response is changed. Also, they show a decreased BDNF receptor expression, which is responsible for neuronal survival and growth. Interestingly, those behaviors were reversible when mice were exposed to a certain bacterium, so Bifidobacterium infantis. Very interesting is that also evolving has been the link between the gut microbiota and sleep. For example, sleep rhythm loss, so like a jet lag, affects the balance of gut microbiota. This dysbiosis, so the loss of balance and rhythmicity, was shown to impact health outcomes. Several lines of research are now evolving, and they pursue the possible underpinnings, so the physiological metabolic underpinnings of the sleep-gut linkage. This research focuses on rodents and adult humans, and babies are much understudied in this field of research, which we found very interesting because in infancy, the gut microbiome just experiences its buildup. And similarly, also sleep regulation is not at all established yet. So that's what we started to focus on in the last few years. In a recent study, we focused on sleep-gut-brain connection in 162 healthy infants and tracked them longitudinally across the first 2 years of life. This highly collaborative approach captured sleep behavior with actimetry, brain activity in sleep with high-density EEG, stool samples to quantify the microbiome maturation and diversity, and behavioral parent-rated scores to capture developmental status. Here we demonstrate that there is indeed a sleep-gut-brain link that develops in infancy. It undergoes transitional dynamics and involves also an early sensitive period. So specifically, the daytime sleep was related to the diversity of gut microbiota. Nighttime awakenings were related to the maturity of gut microbiota and the so-called bacterial enterotype, which is a simplification of classification of gut bacteria. And both sleep and gut microbiota were linked to behavioral developmental outcomes. Strongest connections of that were found at age 3 months. So there's a dynamic interplay of sleep and gut microbiota within healthy human development. And we think as both sleep and gut bacteria are modifiable, they are promising targets also for early interventions in clinical groups. Now what's next? I believe rhythm. We should focus on the development of the two-process model. How does this evolve in humans? And how is rhythm relevant for developmental plasticity and cognition? Also, how does rhythm in the brain and periphery interconnect? Is the regulation bottom up or top down? Also, key transitions should be relevant for future. Where does rhythm evolve? Is that in the fetal stage, in infancy or older ages? Which are vulnerable periods thereof? Which are drivers of the rhythms that support or inhibit rhythm development and thus healthy sleep? Noise, stress, medication, it could interfere with sleep, and some studies show that they don't necessarily have to. What's the role of social context, media exposure, particularly the light of that? What about regularity of bedtimes? Also, last but not least, how is eating and timing of eating involved in sleep regulation? Lots to be done. So finally, maybe instead of compromising on our brain, we should just take care of a good rhythm and a good sleep.